Does everyone hear me well? Okay, so let me move there. Okay, so when we think about multicellular evolution, we're often thinking about the evolution of multicellular organisms. So when we think about the evolution of humans, we might think of an individual human in a population of humans, actually a way smaller effective population size. But we are definitely not thinking about the more than 30 trillion cells in a single multicellular body. And there's good reasons for this. We have evolved a lot of mechanisms to reduce evolvability at the cellular level. And this has led even to the evolution of some cells that basically cannot evolve anymore, like the erythrocytes. But nevertheless, there is evolution within our bodies, and sometimes that evolution, like in cancer, can have major consequences at the multicellular level. And I want to just point out that all these traits are derived traits. So we might think that in the initial steps in multicellular evolution, there's even more conflict and more evolution happening at both levels at the same time. And so I study this multicellular yeast, which is a case of very incipient multicellularity. It evolved in the lab from the Saccharomyces cerevisiae as its unicellular ancestor. And this yeast basically has a life cycle that looks like this. When they divide like other yeasts, except the mother and daughter cell don't separate from each other. So then as they keep dividing, the cluster sort of grows exponentially. And then at some point, it breaks into small propagules that then can grow again. And this mode of reproduction basically already is reducing variation in between the cells in the multicellular organism. So we can imagine that we get that pink mutation, and so it will basically spread on that branch, and then when it divides, we'll have some pink individuals or some gray individuals, but we will not have some pink-gray individuals, which limits competition at the cellular level. And so in this system, I want to investigate how does selection at multiple levels interacts in an adaptive process, and if there are fitness conflicts between the levels, and if selection is capable of sort of aligning the interests at these different levels. And so I choose to work with UV radiation, and there's many reasons for that. One is that it increases the mutation rate, so it actually increases variation at the cellular level. It also, we can think of adaptations at multiple levels, so we can think of a cell adapting by changes in the cell wall, by DNA repair mechanisms. Or we can also think of adaptations at the multicellular level where the external cells are protecting the ones that are in the inside. So then we would expect a change in the size and cell density of a cluster, and we might even expect to see some cellular differentiation when the outside layer is producing sort of resistance to UV, whereas the internal cells should not pay that cost and will not express that resistance. And so first we wanted to test if you could actually see this sort of protection at the multicellular level. So we grabbed different multicellular genotypes and compared them with different unicellular genotypes that had evolved under the same period of time, but without settling selection, so they were not multicellular. And so we can see already that multicellular individuals have higher resistance to UV than the unicellular ones, except for, it's hard to see, but there's a small one here. I'm sorry I'm not using the laser, my hands shake too much. But that small multicellular isolate is so small that it actually doesn't have multiple layers of cells, which actually sort of helps support our hypothesis that what's going on is not just that, oh, you have more cells, so then the probability of a cluster surviving is just the additive probabilities of the cells surviving, but we actually have a non-additive effect that depends on, you need to be of certain size to actually get this effect. Okay, so then we did some selection experiments at 2 of these UV exposure times. So 16 seconds is a time where we get still a lot of multicellular survival, and very few unicellular survival. And then we did an intense selection at 25 seconds, where at 25 we basically see no unicellular survival. And so from now on in my talk, purple or dark reds are gonna be 25 seconds, and 16 seconds is gonna be pink, and I promise the colors are more easy to differentiate than in this plot. So we started with 5 different multicellular genotypes and also a single unicellular genotype as a control. And then we started 3 populations out of each of those 2s. And so some were in a controlled treatment that had no UV. Some started at 16 seconds of UV. And in the middle of the experiment, at 15 days of evolution, we started basically, the controls continue, the 16-second line continues, but we also started 3 more lines that had 25 seconds of UV. And so what are the results? So in this plot, basically we have all the evolved lines, and the gray ones are the controls, the big ones are 16 seconds, the purple is 25 seconds, and they go from like time of evolution. So the light grade one is the ancestral, then 14 days of evolution, 25, in 16 seconds, again, we have 14 days of evolution and 30, and then the 25 seconds, we only have the last time point. And so, as you can see, at 16 seconds, there is some evolution of resistance, and at 25 seconds, especially the 25-second one, gets resistance. But I want to point out that the 16-second line actually over time seems to be doing worse at the 25 seconds of UV. Furthermore, we look at size, and at size we get something really interesting. So these are just 3 of the populations that we had, 3 of the different lines that we started with. And so at 14 seconds of evolution, 14 days of evolution, we get either that the size is maintained or there's even some reductions in size. And 30 seconds, we even see more extreme reductions in size with the lines that evolve either in the control or at 16 seconds. But the 25 seconds, they all increase dramatically in size. And so we focus on these lines. From now on, it's only gonna be the lines that in this population, because it's particularly interesting how it really got a dramatic decrease in size at 16 seconds and a dramatic increase in size at 25 seconds. And this seems to be a conflict between the unicellular and multicellular level at the 16-second lines, where this reduction in size is not just that the clusters are getting smaller, but we get actually an increase in frequency of unicellular phenotypes in those populations. Okay, so then we had this evolved control that was a unicellular ancestor, and so I did competitions against that unicellular ancestor to see sort of what the relative benefits of the multicellular lines were against the unicellular ancestor that had evolved under 16 seconds of UV. And so both the ancestral and the line that evolved on 16 seconds of UV had a similar pattern, where the gray line, which is at 0 seconds of UV, basically, if you expose them, if you do the competitions with 0 UV exposure, you get that the unicellular increases in frequency. So there seems to be some cost of being multicellular to this environment. Then when we do 16 seconds of UV, we get that there's not a significant change in frequency of multicellular or unicellular. And when we do the competitions at 25 seconds, we get an increase of the multicellular isolates. So again, it does seem like at 25 seconds, multicellularity is doing so much better. But then we look at the line that evolved at 25 seconds of UV, and this one was better no matter what the UV environment was, and there was a much more advantage in 16 and 25 seconds. So it seems like basically pushing the multicellular evolution so hard makes it to sort of be able to deal with this trade-off between single cell and multicellular lifestyles. Okay, so then we had this variation, but it was sort of here, at this point we have resistance at the cellular level and at the multicellular level sort of compounded in the same genotypes. And so then we did some crosses to basically get see what the variation was there and breaking the linkage between these two levels. And so we did crosses of the 16 with themselves and 25 with themselves, and then those like F1 lines, we put them again in 0, 16, and 25 seconds of UV only for 5 days, and we again tested for resistance. And so the way this time I tested for resistance was I spot plated them, I put the plates on their UV, and then I basically measured the pixel intensity over time, so taking measures every half hour. So if you have very few survival, you're gonna get a very low pixel intensity because most of the spot is gonna be completely black. If you get high resistance, it's gonna be all white, you have high pixel intensity. And so this is how it looks. So in the upper row is the crosses at the lines that evolve at 16 seconds. On the bottom line is the crosses that evolve under 25 seconds. And so we see that when we do 0 seconds, they all do pretty good. They just grow happily. When we do 16 seconds, in the upper panel, we can see that the ones that then were exposed to 25 seconds sort of increased their resistance, whereas the ones that were at 16 and 0 seconds, we cannot see difference. So that tells that the variation that was available there doesn't get for an increase, like the increase at 16 seconds, it's sort of saturated. You need to push them harder to still see adaptation. But if we see at 25 seconds, we get that there's definitely, the 16 and 25 lines are pretty similar, and those are different from the 0 seconds. And when we push them at 25 seconds, we see a similar but more extreme case of the same thing. And so then I also counted the frequency of multicellular and unicellular colonies in these sexual lines after this competition. And so we can see that this scale, I just want to point out that is a log scale. And so we get that at 0 second, we basically even though these were multicellular isolates, after they went through sex, there were some unicellular isolates, and then after selection, basically most of them become purely unicellular. 16 seconds, we get a low frequency of multicellular isolates, but at 25 seconds, at least in 2 of the 3 replicates, we get that basically all become multicellular and there's no single cells. And so, what does this mean? Basically, you can imagine that there's, at 16 seconds, some sort of fitness peak of being unicellular. And there might be an even higher peak for multicellular, as we saw with the 25 seconds line, that they could do even better. But there seems to be a huge dip in between. Whereas if we expose them at 25 seconds, we are basically not letting any cell survive on their own. It's all the selection is happening at the multicellular level, and that is decreasing the trade-offs between the 2 levels. And what we get there is that we have this sort of landscape where single cells are just dead, and the multicellular ones are doing really well. So quickly, conflicts of selection at different levels can lead to disintegration of these multicellular organisms, so they evolve some sort of unicellular variation. Natural selection can shift the main level of adaptation, sort of aligning the interests. And adaptation at different levels might interact. So the 25-second lines do better than adapted multi or uni at 16 seconds. And so in this system, multicellular adaptation is facilitated by reduced variation within clusters. And with that, I just want to thank my funding sources. Thank you all. Yeah, any questions?